Hi everyone, hope you are all fine. Welcome back to PATH-GENOMICS. PATH-GENOMICS is created to simplify pathology concepts for students, professionals, and enthusiasts alike. Today, we're diving into one of the most crucial steps in histopathology, fixation. We'll cover what fixation is, why it's essential, the different types of fixatives, and some pro tips to get the best results in your lab. So stick around, and let's get started. First, let us know what fixation is. Fixation is the process by which the cells or tissues are fixed in chemical and partly physical states so that they can withstand subsequent treatment with various reagents, with minimal distortion of morphology and no decomposition. Why this fixation is necessary? What will happen if the tissues are not fixed, when it should be fixed? How long it should be fixed? To understand this, we have to learn the concepts of cold ischemic time and fixation time. Whenever a tissue is removed from the body, its blood supply is cut off. Due to lack of oxygen, the tissue will begin to decompose, and this decomposition is directly proportional to the metabolic activity and protein content of the tissues. Tissues with higher protein content generally require longer fixation times because fixatives need sufficient time to penetrate and cross-link with the proteins. This forms the concept of cold ischemic time, which is the time between tissue removal from the body and time of starting the fixation. Delay in cold ischemic time causes significant reduction in the antigen concentration, affecting ancillary studies, while fixation time is the total time that an organ requires for complete fixation. The main aim of fixation is to preserve the tissue as close to their living state possible, to prevent autolysis, to maintain shape and volume of the tissues during subsequent procedures of tissue processing, to harden the tissues, to improve the optical differentiation of cells and tissues. There are some common principles in the process of fixation. After removal from the body, surgical specimens should be placed in fixative as soon as possible. Fixative should be 10 to 20 times of the volume of the specimen. Usually fixation for 8 hours in room temperature is required. 4 hours fixation will be sufficient if done with agitation. Fixation time can be shortened by 25 to 40% if temperature is raised. This makes us understand that there are factors which affects the rate of fixation. Factors affecting fixation. First one is pH. pH of the fixative should be around 6 to 8. High acidity or alkalinity interferes with fixation. Second, temperature. High temperature facilitates fixation. Third, agitation. Agitation increases the rate of penetration of fixative. Fourth, osmolarity. Mild hypertonic, 400-450 mosm, solution is preferable. Hypertonic solution causes cell shrinkage, while hypotonic solution causes cell swelling. Last one is the duration of fixation. Depth of penetration of fixative is directly proportional to the square root of time of fixation. D equals K into square root of T, D depth of penetration, T time duration, K coefficient of diffusion of the fixative. For 10% formalin, K equals 0.78. This means the formalin fixative penetrates less than 1 mm in an hour. The duration of fixation should be the square of the distance the fixative penetrates. For example, formalin and alcohol penetrate the best. There are different methods to fix a tissue. Fixation can be done either in physical way or by using chemicals. Different physical methods of fixation are heat fixation, microwave fixation, freeze drying, freeze substitution, immersion fixation, coating, vapor fixation, perfusion fixation. Chemical method is by the usage of organic or non-organic solvents. When an egg is boiled or poached, it precipitates proteins and solidifies it. Thus heat stabilizes the tissue by cross-linking the proteins. With less distortion of tissues on heating, heat also accelerates other forms of fixation as well as other steps of tissue processing. Next one is the microwave fixation. Microwaving the tissues reduces the fixation time from 12 hours to less than 20 minutes, but microwaving tissues in formalin produces explosive vapors, hence, hood is necessary for extraction of the vapors. Instead of formalin-based fixatives, glyoxal-based fixatives can be used for microwave fixation. Microwave energy interacts with the bipolar molecules, resulting in heat denaturation and disulfide bond formation. It is used for routine histopathology, for electron microscopy, for urgent reporting of biopsies, for rapid fixation. Freeze drying is a type of fixation where tissue blocks are immersed in liquid nitrogen at -40 degrees Celsius, which removes the water from the tissues and acts as a dehydrant. Tissue is then fixed with formaldehyde vapor and processed further. Fresh tissues can be frozen with liquid nitrogen at -190 degrees Celsius, isopentane at -150 degrees Celsius, dry ice, CO2, at -70 degrees Celsius. Freeze fixation is used in cryostat sections. Immersion fixation involves directly immersing tissue samples in a fixative solution to preserve their structure and cellular components for subsequent processing and analysis. Coating fixatives. Coating fixatives are substitutes of wet fixatives. Aerosols are applied by spraying the smear immediately after preparation. Example, Carbowax fixative. The distance from which the slides are sprayed with an aerosol fixative affects the cytological details. 25 to 30 centimeters is the optimum distance for aerosol fixation. Perfusion fixation is a method of tissue preservation where a fixative solution is injected into the circulatory system of the specimen to distribute the fixative throughout the tissues, ensuring better preservation compared to immersion fixation. These were the different physical methods of fixation. Now let us see about chemical fixatives. Chemical fixatives are of three types: coagulants, crosslinkers, and compound fixatives. Coagulants precipitates the proteins, crosslinkers crosslinks the proteins with nucleic acids. Compound fixatives does both the function. Chemical fixatives are classified into simple and compound fixatives. Simple fixatives are formalin, mercuric chloride, osmic acid, picric acid, acetone, ethyl alcohol, osmium tetraoxide, osmic acid. Compound fixatives are the combination of more than one simple fixative. They are further classified into microanatomical, cytological, and histochemical fixatives. Microanatomical fixatives are formal saline, neutral buffered formalin, Zenker's fluid, Buon's fluid. Cytological fixatives are further classified into nuclear and cytoplasmic fixatives. Nuclear fixatives is Carnot's fluid. Cytoplasmic is Champy's fluid. Histochemical fixatives are cold acetone and ethanol. Here ends the session on basics about fixation. Let's us learn about each fixative in further videos. Like, share and subscribe for more videos. Let us meet next time and learn about the fixative formalin in detail. Until then, keep exploring, keep learning, keep looking deeper. Every slide tells you a different story. Stay curious and keep those microscopes busy. Goodbye. Happy diagnosing.